Hi, everyone. I think we'll go ahead and get started. So welcome to the Department of Medicine Grand Rounds this morning. We have a fantastic talk prepared by Dr. Tague this morning. So just some brief information about her. So she got her medical degree at the Northwestern University School of Medicine, followed by residency at Loyola University Medical Center. She then came here for Pulmonary and Critical Care Fellowship at Washington University School of Medicine, where she also got a Master's of Science in Clinical Investigation. She's currently an assistant professor of medicine within the Division of Pulmonary and Critical Care Medicine, and her research interests include the pharmacogenetics of lung transplantation. She has a fantastic talk today entitled The Aging Lung, Implications in Advanced Lung Disease and Lung Transplantation. Let's welcome Dr. Tague.
Thank you, and thank you all for coming. I'm excited to share this talk with you all today. So we'll go ahead and get started. I have no relevant disclosures for this talk. I want to start with the general overview of aging, and the reason this is so important, I think, is because we're an aging nation. By 2034, we'll have more adults over the age of 65 than we do children under the age of 18. And by 2060, 1 in 4 Americans will be over the age of 65. And it's not just proportions, it's also numbers. In 1960, we had about 10 million adults total over the age of 65. By 2060, that'll be almost 60 million. And so we are an aging population, and along with aging comes diseases associated with aging. And I'm going to talk about a couple of those diseases today. Aging at its core is a time-dependent functional decline that is mediated by the accumulation of cellular damage. And in 2013, borrowing from the cancer literature, these 9 hallmarks of aging were proposed, and they included altered intracellular communication, genomic instability, telomere attrition, epigenetic alterations, loss of proteostasis, deregulated nutrient sensing, mitochondrial dysfunction, cellular senescence, and stem cell exhaustion. For the purposes of today, we only have an hour, so I'm going to focus on genomic instability, and I'm going to argue that telomere attrition is a subset of that, and also cellular senescence as an endpoint. So I want to start with genomic instability. So we are constantly bombarded by insults to our genome. These include exogenous environmental insults, UV radiation exposures, chemicals, chemotherapeutic agents for a lot of our patients, as well as endogenous insults. And that includes reactive oxygen species generation, replication errors, and errors in our own internal sort of DNA maintenance mechanisms. And our DNA has to withstand these insults and remain competent. And we have mechanisms that allow us to do that. Now, I won't go through the details of this. I just want to point out that when DNA damage occurs, there are 2 main pathways by which repair proceeds. And the main mediator of that, as I'm sure a lot of us are familiar with, is P53, which is sort of a master regulator of DNA damage repair. This ATM check 1-2 pathway into P53 allows for damaged cells to exit the cell cycle. So to prevent proliferation and replication of damaged cells, they enter a cell cycle arrest whereby DNA damage repair can occur. Additionally, this P16 alternative pathway also leads to the arrest of the cell cycle in damaged cells through the cyclin D and retinoblastoma protein. And I also want to point out an important regulator of these 2 main pathways is PPM1D, which serves to inhibit both P53 and P16. And so it is involved in both of the major pathways by which our damaged cells undergo repair. So at homeostasis, when we have a DNA insult or injury or damage, our DDR activation occurs, our DNA damage response activation occurs, and those cells exit the cell cycle while repair can occur. When we have imbalance or we lose homeostasis in our DNA damage repair mechanisms, or we have defective DNA damage repair mechanisms, we get accumulation of DNA damage, accumulation of cells with damage, which leads to the production of reactive oxygen species, leads to a pro-inflammatory phenotype, activation of the inflammasome, and release of pro-inflammatory cytokines, which causes tissue and structural damage. And so regulating and maintaining homeostasis in our DNA damage repair is crucial and is one anti-aging mechanism that we have. Another mechanism of aging is the telomere biology disorders. And the telomeres, we all know, are sort of the caps on the ends of our chromosomes. Their essential function is to protect the chromosome end. If there were no telomeres, our chromosome ends would be recognized by our DNA damage repair mechanisms as a double-stranded DNA break, and that would be inappropriately activated. And so our telomeres are there to protect our chromosomes, the ends of our chromosomes. And they are a nucleoprotein complex. They are DNA repeats, but there's also telomeric RNA associated with the telomeres, as well as these protein complexes whose essential functions are to preserve telomere length, structure, and function. Now, our DNA replication is semi-conservative, and what that means is that the ends of our chromosomes don't get fully replicated when a cell undergoes division. And so telomeres actually progressively shorten as a natural consequence of aging. And our maintenance mechanisms make sure that that is a slow and process such that our telomeres are maintained throughout the course of our life. When a telomere gets critically shortened, in other words, when it's too short to maintain that critical function, that cell actually enters a state of senescence. It exits the cell cycle, it no longer proliferates, and it actually undergoes a controlled apoptosis pathway. When you have defects in any of these telomere maintenance genes, either a maintenance defect or a defect causing instability of the telomere structure, you can get accelerated loss of telomere, so accelerated telomere shortening occurring in, say, the 3rd or 4th or 5th decade of life as opposed to the 6th, 7th, or 8th decade of life. Or you can get sudden telomere loss due to instability of the telomere structure. And when that occurs, you get dysfunctional telomere maintenance. You get early and increased immunosenescence, and you get that imbalance of the DNA damage repair and loss of that homeostasis, leading to essentially early loss of cell proliferation as well as early loss of renewal capacity. And there are some pathological states or diseases that are very well associated with telomere biology disorders, one of which I will get to in more detail later is pulmonary fibrosis. What I want to do is introduce a different type of genomic instability that was not included in that original hallmarks, and that is clonal hematopoiesis. So clonal hematopoiesis is well known likely to our hematology and oncology colleagues as the accumulation or the occurrence of somatic mutations in hematopoietic cells. And that's a normal process that happens with aging. It is primarily a disease associated with aging. The prevalence goes from about 10% in those under the age of 40 to upwards of 30 to 40% in those over the age of 80. And what happens is these somatic mutations occur. There's acquisition of a somatic mutation that infers some sort of selective fitness advantage. And when that happens, that mutation in that cell expands into a clonal population, all harboring that mutation. And this happens in association with aging in a very predictable manner. There are routine genes that are involved in the development of age-related clonal hematopoiesis, or what is termed clonal hematopoiesis of indeterminate potential, or CHIP. And these genes are involved in epigenetic modification or regulation, as well as in the DNA damage response. So this is an investigation looking at the prevalence of clonal hematopoiesis in a general population. This was using a UK Biobank sample, and you can see that by large, the most commonly associated genes with CH is DNMT3A and TET2, which are both epigenetic modifiers. And then subsequently, you have AXSL1, PPM1D, TP53 and ATM, which are involved in the DNA damage response. And as you can see, no matter the associated gene, CH or CHIP is positively associated with age. So it increases with age across all mutations and overall. And this is important because different patterns of CH are associated with different disease states. And so our epigenetic modifiers have been shown to predispose to AML. They've also been associated with a pro-inflammatory state that leads to cardiovascular disease. Our DNA damage response, CH, is usually associated with therapy-related malignancies, with chemotherapy resistance, and is not normally considered a prevalence in sort of age-related CH. And then what I'm going to talk about today is how CH is involved or manifests in pulmonary disease as well. And not only do the different patterns of CH predispose or associate with different patterns of disease, but different exposures cause different types of CH patterns. So this is another study led by Kelly Bolton, actually here in a division of hematology, where they looked at the prevalence of clonal hematopoiesis in patients who had undergone some sort of chemotherapy. And what you have here, as you can see, the most commonly associated or mutated genes are, again, those epigenetic modifiers, DNMT3A, TET2. But what I want to point out is that the risk factor for the development of CH alters the pattern in which CH develops. And so when we look at age, you see a prevalence of splicing mutations versus when you look at therapy-associated CH, so patients who have received some form of chemotherapy, you see much more commonly CH in the DNA damage response genes versus smoking, where again you see a combination of DNA damage response and splicing factors. And so the pattern, the exposure changes the pattern of the development of clonal hematopoiesis as well, which again lends itself to the development of different disease states. And what's more, there's a direct relationship between clonal hematopoiesis and telomere length. So there are two distinct mechanisms of genomic instability, but they are actually intricately related. So this was a study looking at both the top med and UK Biobank cohorts, so a total of over 100,000 subjects, and they measured essentially clonal hematopoiesis of indeterminate potential, or CHIP, as well as estimated leukocyte telomere length. And first performed just a regression analysis to look at the association between the two. And what they found was that the development of CH overall, which is this first column here, was associated with a shortened telomere length compared to patients who did not have CH. Additionally, when you looked at gene-specific CH, genes involved in DNA damage response, as well as JAK2, were associated with significantly shorter telomere lengths than compared to overall the CH distribution. Additionally, the burden of CH, or the number of mutated genes, as well as the variant allele frequency of the prevalence of that mutation, were negatively correlated with telomere length. In other words, the higher your burden of CH or CHIP, the shorter your telomeres. Next, in looking at Mendelian randomization of these two to essentially look at causal inference of these two, when you look at leukocyte telomere length as the exposure and you look forward to the development of CHIP, we see a positive correlation between longer telomere length and the development of CHIP. And on the converse, when you look at CHIP as your exposure and telomere length as your outcomes, you actually see that patients who have CHIP have shorter telomeres when telomere length is considered the outcome. And if you think about it from a mechanistic standpoint, that makes sense because longer telomeres means the ability to withstand more replication or replicative stress. And so that allows for more opportunities for the development of these somatic mutations and more opportunities for the development of CHIP. Whereas once you have the development and expansion of these clonal populations, that represents an imbalance and a switch in the ability to maintain your DNA damage repair mechanisms, and so you have an accelerated telomere shortening. And one of the ways that in which this is integrated is the endpoint of cellular senescence. So cellular senescence is a stable arrest of the cell cycle. These cells are not dead, actually. And this occurs, it's a normal process of development. It occurs in embryonic development. It's essential in maintaining tissue homeostasis. When we're younger, we have robust repair mechanisms that allows for the repair and the withstanding of the damage that our genome undergoes. And these are both anti-aging and anti-malignancy mechanisms, because those damaged or mutated cells are not allowed to proliferate. As we get older, we get the accumulation of cellular damage, we get decreased efficiency of our repair mechanisms, and that leads to increased cellular senescence. And additionally, we get decreased clearance of these senescent cells, and we have a decreased renewal capacity. And that's important because these senescent cells are not dormant. Senescent cells have a very well-defined phenotype that is a pro-inflammatory phenotype. Called the senescence-associated secretory phenotype, or SASP. And these cells actually release pro-inflammatory cytokines and mediators such as IL-6, IL-8, and they promote autocrine and paracrine dysfunction, leading to tissue dysfunction, increased inflammation, and effects on adjacent cells. Inflammation, when this occurs in immune cells, it leads to immunosenescence, which I'll touch on in a second. And additionally, stem cell exhaustion, because we have depleted capacity for cell renewal as we age. And that overall leads to the progression of aging. Senescence in the immune system. So as we age, naturally, we have decreased immune competence. We have decreased adaptive immune function. Our T-cell receptor repertoire is decreased. Our B-cell antibody diversity decreases, and we have restricted or overall impaired response to antigenic challenge. Additionally, we have persistent innate immune activation. So we have increased infiltration of neutrophils and macrophages, increasing levels of pro-inflammatory cytokines circulating, and we get this inflammation, this so-called inflammation, where we get inflammasome activation and inflammasome-mediated cytokine production, which again leads to the release of these pro-inflammatory mediators. And this inflammation actually increases our susceptibility to common infections, particularly latent viral infections like cytomegalovirus or CMV, as well as varicella zoster, which is why we see reactivation of varicella in older adults specifically. And so I will say that senescence is at its an integrated endpoint of aging. No matter the mechanism of the damage that occurs, the endpoint in which these cells exit the cell cycles to attempt to repair the damage leads to cellular senescence. And when we age, or as we age, the burden of senescent cells increases and causes the damage that we see associated with this SASP pro-inflammatory phenotype. And so what we really have is an integrated model of aging, rather than these unique or distinct slices of the aging pie, so to speak. We have primary mediators of aging, such as genomic instability and the subsets of that, leading to responses to that primary damage, which is mitochondrial dysfunction, reactive oxygenation species production, cellular senescence, and overall leading to decreased immune competence, decreased capacity for proliferation, increased stem cell exhaustion, and aging. And so rather than having distinct factors, we really have this integrated model of aging whereby all of these factors contribute to varying points or to varying extents to aging and to different disease processes. And it's even not this neat little circle, but there's all these interactions between these different mechanisms. And so looking at how these mechanisms affect advanced lung disease, in particular, I want to focus on two specific types of lung disease which are associated with aging, and that is COPD and pulmonary fibrosis as a representative of the larger sort of group of interstitial lung diseases, all of which have a final phenotype of fibrosis. These are two of the most common chronic lung diseases that we manage, and that are the two of the most prevalent lung diseases in our population. And in addition, they are the two most common indications for lung transplant. Upwards of 70 to 80% of our lung transplant recipients are transplanted for one of these two diagnoses. And so understanding how sort of the underlying pathogenesis of these diseases, understanding how aging and the genomic instability contributes to not only the pathogenesis of these diseases, but also the progression of these diseases to transplant is essential. I want to start with aging or telomere biology disorders in IPF. IPF, I mentioned earlier, was one of those sort of diseases that are pathognomonic with telomere biology disorders. And this is a study of IPF recipients and looking at essentially defects in these different telomere maintenance genes and identification of germline defects. And almost half of patients with IPF have an identifiable defect in these germline defect in these telomere maintenance genes. And that is important because we also know that defects in these telomere maintenance genes are also associated with progression of disease. And that this is a study looking at lung transplant recipients, of IPF patients who progress to lung transplant. So these are about 150 lung transplant recipients versus 282 non-transplanted IPF patients. And if you look at the prevalence of telomere maintenance gene germline mutations in these patients, you can see that 1 in 4 of patients who progress to lung transplant have an identified mutation in a telomere maintenance gene versus only just under 12% of those patients who don't progress to lung transplant. And they have significantly shorter telomeres. So the red is patients who progress to lung transplant. The black are those who don't. They have significantly shorter telomeres. And when you look at the relationship between telomere gene variants and telomere length and progression to lung transplant, an interesting thing falls out. Telomere gene variants alone is not significantly associated to progression to lung transplant, but telomere length itself is, and particularly telomere length less than the 10th percentile. And what that tells me is that the germline mutations in the telomere maintenance genes don't tell the whole story. There are other factors that are contributing to telomere shortening or decreased telomere length that beyond these germline mutations. Telomere biology disorders in COPD is something that's not talked about as much, but that is also not a new concept. This is actually a 2009 study that was published in the Blue Journal that looked at 50 patients with COPD and matched them for age, sex, and smoking history to patients without COPD. And what you can see is that patients with COPD had significantly shorter telomere lengths overall, and in particular, a significantly higher proportion of them had telomere lengths in the bottom tertile, or bottom 33% of length. And this is looking at COPD patients versus controls, significantly shorter telomere length. And when you look at it even by age, across all age groups, this is less than 60, 60 to 69, and then greater than or equal to 70. The gray boxes are the COPD patients, the white boxes are the control patients. And so across the age spectrum, patients with COPD have significantly shorter telomeres, and it's associated with functional outcomes. So this is PaO2 and PaCO2, and you can see there's a positive relationship between PaO2 and telomere length and a negative relationship with PaCO2. So longer telomeres are associated with a better physiological profile in patients with COPD. When we turn to clonal hematopoiesis, this is a newer area of investigation as it relates to lung disease, and there's not much data out there. This is a study that we published last year looking at the prevalence of clonal hematopoiesis in patients who underwent lung transplant. We sequenced pre-transplant samples from these patients to assess the burden of clonal hematopoiesis, and what we found is that of patients with IPF who underwent lung transplant at our institution, almost 80% of these individuals had an identifiable mutation or CH mutation, and that's any CH, so all of those genes that we talked about previously. And furthermore, over 50% of these patients had an identifiable CH mutation in a DNA damage response gene. 50%. If you recall back to COPD and IPF, we were thinking, and those sort of UK Biobank and Top Met cohorts, the prevalence of DNA damage CH in those cohorts were around the 10% mark. And so the lung transplant recipients are enriched for these DNA damage response genes. And you can see, again, you see the normal patterns of DNMT3A being the most common, but just as prevalent is PPM1D, which is one of those regulators of the DNA damage response. When we turn to COPD, there's been a little more investigation of colon in COPD. This was a study published a couple of years ago looking at 4 different COPD cohorts, a total of just under 3,000 patients. And again, they're looking at the prevalence of CH. This is a cross-sectional study looking at the prevalence of CH in these patients. And what they find is patients in each of these, across these COPD cohorts, we see the normal sort of age-related or age-associated patterns in COPD or in colon which is epigenetic modifiers, DNMT3A and TET2, much less prevalent, hovering around anywhere from 2 to 10% in these patients, is mutations in DNA damage response, such as PPM1D. And when you look at CH association with the severity of disease, so these are patients who had GOLD class or 2 to 4 and 3 to 4, you see a positive association overall among all these cohorts of the presence of CH with severity of COPD disease. And furthermore, when you look at FEV1, so sort of functional measure of the severity of CH, patients with CHIP overall among these cohorts had significantly lower FEV1 than patients without CHIP or without CH. And so what does this mean in terms of disease severity or disease progression? Well, again, we looked at patients with COPD who progressed to transplant. So this is pre-transplant CH assessment in COPD patients who progressed to transplant. And again, what we see is a startling almost 90% of our patients with COPD who progressed to transplant had at least one CH mutation identified. And the DDR-CH, again, was about 12 to 14% in these patients. And again, you see those epigenetic modifiers, DNMT3A, TET2, being the most common. But again, the third most commonly mutated gene that we found was PPM1D. And so what does that mean? We have an accumulation of these DNA damage response mutations in our transplant patients. What does that mean for transplant? Why is it important? Well, just like in the general population, our transplant recipients are getting older. Our median age of a transplant recipient was about 55. It's now just over 60. And additionally, the distribution of indications for transplant are changing. So, you know, in the decades prior, the most common indications for transplant, COPD and cystic fibrosis. By far, the most common indication for transplant now is interstitial lung disease and IPF being the representative disease for those groups of diseases, of pulmonary fibrosis diseases. And those represent almost half of the lung transplant recipients with in the current era of transplant. Additionally, although the frequency of transplant for COPD is decreasing, the age at which patients undergo transplant for COPD is increasing. So the age of transplant, even for COPD patients, has increased from early to mid-50s to over 60 in the current era. And so overall, we have an increasingly older transplant population. And so they have an increasing burden of these age-related changes in our immune system, and increasing accumulation of cellular damage and genomic instability. And then on top of that, we put them through a transplant. And the transplant process itself is associated with impaired insults to the immune system. And so even from the beginning, our transplanted organs undergo this ischemia reperfusion from the beginning. So as we go through organ procurement from the donor to the recipient, our organs undergo a period of ischemia, and that ischemia produces all types of derangements, increased reactive oxygen species and hypoxia leading to mitochondrial dysfunction, increased leukocyte activation and recruitment into the allograft, upregulation of surface molecules leading to altered intercellular communication. We get loss of proteostasis because of impaired autophagy, and we get injury related to reperfusion, all of which cause primary dysfunction in the allograft, and all of which can lead to a state of immunosenescence. And so understanding how lung transplant can, or the process of lung transplant can exacerbate all of these mechanisms of aging and accelerate it is important in understanding the biology of the allograft function. And so looking at these mechanisms of genomic instability within the lung transplant population, this is a study out of UCSF where they looked at telomere biology disorders and telomere length and the association with lung transplant outcomes. And what you can see here is primary graft dysfunction, or PGD, which is graded from 0 to 3, with 3 being the most severe, is that severe primary graft dysfunction is associated with significantly shorter telomere length. And additionally, chronic lung allograft dysfunction, or chronic rejection of the allograft, is associated with significantly shorter telomere length. And those patients with the shortest telomere lengths here in red have a significantly decreased chronic lung allograft dysfunction-free survival. And when they looked at, when they performed differential gene expression studies in these different subsets, shorter telomere length and development of rejection was associated with a pro-inflammatory phenotype. And that links our telomere biology disorder in the post-lung transplant populations with functional outcomes in transplant. And so our next question was, okay, how can we investigate other avenues by which these mechanisms of genomic instability can contribute to derangements in the immune system post-transplant and post-transplant outcomes? And so together with Karee Etchen and Dan Lincoln in the Division of Hematology, as well as Andrew Gelman in the Department of Surgery, we actually investigated clonal hematopoiesis in our lung transplant population here. So we performed error-corrected sequencing using a custom gene panel of 85 adult lung transplant patients, 20 of which we had prospective longitudinal data on, and we compared their prevalence and distribution of CH with healthy older control patients who were age matched. Our cohort looked pretty similar to our larger lung transplant cohort. Just over half were transplanted for ILD, another 30% for CF, and so this represents about 80% of our cohort was represented by ILD and COPD. These patients had about 2/3 had pre-transplant cigarette use. The vast majority underwent bilateral transplant, and then they had a standard maintenance immunosuppression regimen. And what we found was very interesting. So walk you through this. The gray is the control group. So these are our control adults. And the red represents our transplant patients. We divided them into interstitial lung disease, COPD, and then other diagnoses. Up here, they're organized by age. Each column here represents an individual patient. And so what we see is when we look at the patterns of clonal hematopoiesis in these patients, what we see is our control groups have what we would expect the typical CH patterns associated with age. By far, DNMT3A is the most common, and they have a couple of mutations in our SRCAP and ZNF318. They have no mutations in DNA damage genes. You compare that to our lung transplant recipients, which again, by far the most commonly mutated gene is DNMT3A, consistent with age-related patterns, but also we see a unique and distinct signature of CH in DNA damage response genes, and particularly in PP-M1D in both ILD and COPD patients, and also some in TP53 and ATM. And so there's a distinct pattern within these lung transplant recipients of increased DNA damage response CH. Additionally, and this is significantly different, so when we group them by epigenetic modifiers, which is this first column here, and DNA damage response, which is the second column here, versus others, our transplant recipients are enriched for DNA damage response genes. When we looked at factors that were associated with DNA damage response, CH, we predictably saw that age was associated, as well as pre-transplant cigarette use, and a trend towards increased prevalence or increased likelihood in patients with ILD. And when we looked at our prospective subgroup of this, so these are patients, we have pre-transplant samples, and we followed them prospectively through a certain period of time, up to 6 months for both, we see persistence of this pre-transplant DDR-CH in our prospective cohorts as well. And we looked at factors that were associated with DDR-CH versus NECH. We evaluated diagnosis. Again, as I said, DDR-CH was more common in ILD patients. But looking at other pre-transplant factors, there was no relationship with pre-transplant immunosuppression use, mycophenolic acid, or other potentially agents that can exert a selective pressure, steroids, or even pre-transplant pirfenidone and nintedanib for our IPF patients. Next, we looked at the relationship between telomere biology disorders and clonal in our transplant population. And so this is a subset of patients who either had ILD or who had an identified DDR-CH mutation in our sequencing panel. And we performed whole exome sequencing looking for germline mutations in these telomere maintenance genes. And we found a subset of these patients. In fact, we were able to identify germline mutations in telomere maintenance disease as well as surfactant diseases. The next thing I wanted to do is look at outcomes related to transplant, because the whole point of this is to how does this relate to functional changes in our lung transplant recipients. And what we found was that DDR-CH in particular was associated with a significantly increased risk of CMV viremia. So what we have here is red represents patients with DDR-CH, blue is patients with non-DDR-CH, and then black is patients with no CH. And so our DDR-CH patients had significantly higher rates of CMV viremia when compared to both patients with non-DDR-CH and patients with no CH. And when we looked at this in logistic regression analysis adjusted for CMV risk status, so donor versus recipient CMV status, as well as age, we see that this relationship persists. DDR-CH is significantly associated with an increased risk with an adjusted odds ratio of over 7 of CMV viremia. And look at overall survival in these patients, DDR-CH was associated with a decreased CMV CMV viremia was associated with a decreased CMV-free survival in these lung transplant recipients. And this is important because CMV is a major source of morbidity in our lung transplant recipients. It represents an impaired or defective adaptive immune response, in particular CD8 positive T cells. And it's associated with poor outcomes in lung transplant recipients. When we look at CMV serostatus between donor recipients and age and the association with outcomes, we see that patients who are high risk, that is a negative serostatus recipient and a positive serostatus donor, have significantly poorer survival than patients who are low risk, where the recipient and donor are both negative. And this is despite the fact that we, in these high-risk patients, we actually provide CMV prophylactic therapy to them post-transplant. And so despite that, these patients still have poorer survival. But what's even more interesting is when you look at this by age, this relationship appears to be solely seen in patients who are over the age of 60, which is representing more and more of our lung transplant population. Patients under the age of 60, even despite CMV mismatch status, there's no significant difference in survival. But when we look at patients over the age of 60, there's an adjusted hazard ratio of death of 4.5. And so this really seems to be related to an interaction between the recipient's age in addition to CMV serostatus. And so what role is the aging immune system playing in this impaired CMV immunity, and why is it impacting post-transplant outcomes so much? And one of the reasons is that impaired CMV immunity or reactivation of CMV viremia is indicative of overall decreased immune function, and it has been associated with genomic instability and those other mechanisms of aging. And so this is a study out of Penn, where they looked at impaired CMV immunity and the relationship with short telomeres in IPF lung transplant recipients. And what they see, they have 42 patients with and without IPF, so 84 total. And they look at CMV controllers, so patients who have no persistent CMV viremia, versus CMV relapsers, who have patients with recurrent CMV viremia, where the relapsers are in red. And you can see here that the relapsers have significantly shorter telomere lengths than the controllers, where 80% of the relapsers have a telomere length at or below the 10th percentile. And when you also look at CMV-specific proliferative responses, this bottom panel here, the IPF patients with the very short telomeres, or in the bottom 10th percentile, in the orange here, have significantly decreased proliferative responses in response to CMV-specific antigen stimulation. And they also have decreased cytokine responses, so decreased interferon gamma, decreased TNF alpha. And so you overall get an impaired CMV immunity related to overall an aged immune system represented by short telomeres. When you go a step further and you look at the impact of immunosenescence, so this is, again, a study of UCSF where they looked at shortened telomeres, age, and the development of immunosenescence defined by senescent markers, CD57, CCR5. They found, and they did this prospectively, they found that the transplant process itself was associated with prospective change in the distribution of T-cell phenotypes, whereas pre-transplant, there's no difference between patients with normal telomeres, NT in the red, and short telomeres in terms of the distribution of the different T-cell types, naive, effector, and effector memory. But as early as 6 months post-transplant, the short telomere patients have a significantly increased number of terminally differentiated T cells, and that is further increased at 12 months. And so you get a change in the distribution of T cell phenotypes that you see post-transplant. Additionally, patients with shorter telomeres had a significantly, and this is specifically in the CD8 T cells, they have a significantly higher number of cells that are in a senescent state, where they express a senescent phenotype, and that is a manifestation of the transplant process. So this is not present pre-transplant in patients with short telomeres, but as early as 6 months post-transplant, you have a significantly increased proportion of CD8 positive T cells that are in a senescent state and have a senescent phenotype, and that's independent of the age of the transplant recipient. So even the younger transplant recipient's age is not protective for them in the post-transplant state. And so you have this link between aging, telomere length, and outcomes post-transplant, but again, telomere length by itself doesn't explain all of this difference. And so we are investigating further right now, looking at clonal hematopoiesis as a potential link between these factors of genomic instability that can explain perhaps some of this unknown genetic contribution to the development of these diseases. So in particular, clonal hematopoiesis in the DNA damage response genes, PPM1D, ATM, and TP53, whereas in a normal sort of pathway of DNA damage response, you have telomere shortening, environmental exposures like cigarette smoke leading to a DNA damage response, which leads to a senescent phenotype. We are purporting that there is a mediation effect with clonal hematopoiesis, which bidirectionally affects the DNA damage response and also mediates the activation of the sort of master regulator of DNA damage response in P53, and in particular in PPM1D as a major inhibitor of activation of this pathway. And so where we are now is we're looking to further elucidate the mechanistic link between clonal hematopoiesis, telomere biology disorders, and immunosenescence. And our goal is to, in particular, look at post-transplant immunosenescence, and specifically in CD8 positive T cells, as they play such a key role in post-transplant immune competence, as well as post-transplant allograft tolerance. Additionally, identifying how CH plays a role in advanced lung disease, the disease progression or disease phenotype can help us further refine the phenotype of these advanced lung diseases, help us further understand who will have disease progression to deteriorating lung function leading to lung transplantation. And then within our transplant community or population, looking at how we can use this information to personalize our approach to transplant management, particularly immunosuppression management, adjusting intensities based on not only telomere length, but also patterns of CH and immunosenescence, as well as refining our prophylactic therapeutic approaches, particularly for CMV. And I will stop there. That's all I have for you. I'd like to acknowledge the members of my lab who helped perform this work, as well as Dr. Gelman and Dr. Link in his lab, our entire lung transplant group, our amazing lung transplant patients, without whom this wouldn't be possible, McDonald Genome Institute, who performed all of our sequencing for us, and the generous organizations who have funded this research. And I will stop there, and I'm happy to take any questions.
Dr. Tague, that was a great talk. Thank you so much. We have time for questions. Steve, Brody?
Great talk, Laneshia.
Thank you.
You focused on germ cell mutations, but do you or other people also find any of these in the cells, for example, structural cells of the lung or resident immune cells?
Yeah, so as I mentioned, CH is in its infancy. Oh, sorry, the question was, I talked mainly about immune cells, and the question was, have people investigated CH in other structural lung cells or tissue-resident lung cells? So I mentioned CH is in its infancy, and so it hasn't really been investigated. It's really an area within the realm of lung disease, advanced lung disease, that is wide open. But what some people have looked at is actually telomere length within lung epithelial cells, particularly, and its association with primary graft dysfunction. And they have identified a link within lung epithelial cells of shortened telomeres and increased risk of primary graft dysfunction as well. So I would think that as we further investigate and elucidate this, we'll see that sort of those local tissue resident cells also contribute to the phenotypes that we're seeing.
There's a lot of smokers that have no cardiac disease. Do you think that there's something specific in your population that has to do with this exclusion? Or if you took other transplants, say heart transplant, are you going to get the same sort of thing?
So I think it's probably our lung transplant in particular. The question was, is this a unique phenomenon within lung transplant or other transplant populations having the same exposure? And for clonal hematopoiesis in particular, I think our lung transplant recipients represent a unique population, not only because of what you just said, but also because the lung undergoes continual exposure to environmental antigens. There was actually a study of CH in heart disease and recipients who progressed to a heart transplantation, and they did not find the same patterns of DNA damage response, CH, that we see in our lung transplant recipients. They found patterns associated with those epigenetic modifiers, which have been extensively studied actually within cardiovascular disease without transplant, and are associated with the pro-inflammatory phenotype leading to increased risk of atherosclerotic disease and other cardiovascular diseases as well. So I think this is a phenomenon that's unique to lung transplant recipients. Thank you. Great question. So the question was, is CH causative to the lung disease, or if there is the shared risk? I think it's probably the latter. There have been a couple of Mendelian randomization studies now looking at causal inference between CH And I showed the one on short telomeres, also looking at progression or development of lung disease. And it seems that there is a bidirectional relationship, such that CH represents sort of a tipping point in an overload of our DNA repair mechanisms, and it leads to accelerated decline in function once that occurs.
Great talk, thank you. I was interested in the slide you showed on the demographics for the changes in lung transplantation over a few decades, and noticed weights going up and BMI, and you may have mentioned this, but that just a reflection of society and playing a direct role. And I also noticed a decrease in the number of females that were being transplanted. Just wondering if you could comment.
Yeah, so the question was about changing demographics in lung transplant. One of the main driving factors, well, a couple of things. First, there was a change in how we allocate allografts, such that the scoring system that we use was changed to try to decrease waiting list deaths. But what that did was it increased the scores for patients with IPF significantly. And so we see a lot of IPF and ILD patients being transplanted now. Those patients have a male predominance. IPF has a male predominance in general. And so that accounts, I think, for the increase in the number of males. Additionally, with the change in that lung allocation score, as well as with the advent of new meds for cystic fibrosis, delaying and actually controlling that disease without the need for transplant, we have seen a shift towards older adults who tend to weigh more versus younger CF patients who tend to be very thin. And there's also been some leniency in the BMI cutoffs for transplant as well, I think probably as a reflection of the general increasing obesity epidemic in our society.
Laneshia, thanks so much.
Thank you.